Thank you so much for coming by for this collab session. So as a brief introduction, I'm one of the co-founders of Wisdom and Senior Director of Scientific Affairs. So for today's collab session, I'm going to talk about two topics. First is to sort of really give a walkthrough of Wisdom's platform technology called Merscope, what it does and how it works. And then in the second part of my talk, I'm going to focus on explaining how to use Merscope platform to do in situ single-cell transcriptomic imaging in FFPE sample types. So if you have questions later, feel free to ask at any time. So, all right. So just to give some context and background first, as we all know, biological systems are very complex and made of many different cell types organized in 3D, and they express genes quite differently. And over the past few decades, sequencing has really transformed our way of understanding different cell types and how they express genes. But sequencing still has fundamental limitations. That is, it's difficult to know where cells are coming from because you need to dissociate or destroy the cells for sequencing. Thereby, it can only show us parts. It's very difficult, for example, to use sequencing to infer how cell interact with each other or where cells are coming from, right? And only in recent years, this new field called spatial genomics now started to come out and aims to add the critical spatial component back. And now we're having the true ability to do a highly multiplexed direct in situ detection in tissue samples. So as a company, Wisdom is really the first one that has brought up a true single-cell spatial genomics platform called Merscope to the market. This is an RNA imaging-based platform that can profile hundreds to thousands of different genes in a single run with extremely high sensitivity and accuracy. And this platform can capture very large imageable areas up to 1 square centimeter sized tissue down to 100 nanometer resolution. And there's no need for sequencing. So Merscope is built based on the foundational technology called MERFISH, which stands for multiplexed error-robust fluorescence in situ hybridization. So MERFISH leverages single molecule FISH's direct in situ RNA quantification capability, and combined with a proprietary barcoding system, it now enables massively multiplexed RNA imaging in biological samples. So to work with Merscope, it usually begins by a custom gene panel design, and as shown on this slide, we do have a gene panel design portal here where you can pick your genes of interest. And for now, we offer up to 500 genes, and there will be up to 1,000 genes coming very soon. Then with the probes design, then the sample will be hybridized with the probes and undergoes to several steps of sample prep, and then loaded into the flow chamber. And a corresponding reagent pack will be selected for this experiment. Then the sample will be loaded to Merscope machine, and then automatic data acquisition happens. And then the sample will start to go through multiple rounds of imaging, and then we will later decode the images. And then downstream with the decoded data, we will be able to visualize the data in the visualization software as shown here. Working with MERFISH involves 3 key stages. As shown here, in stage 1 is to hybridize the sample with these tens of thousands of unique encoding probes containing the binary barcode. Then the sample will be sort of gel embedded and cleared to remove anything that could interfere with the measurement. Usually removes and reduces a lot of autofluorescence. And then in the final stage, then the readout probes will flow in, and then through multiple rounds of imaging, it will generate an optical barcode, basically fluorescent on and off patterns for individual genes. And essentially what you will get is the ability to visualize the genes in situ as shown by the picture on the right, where each colorful dot is indicating different RNA transcripts. So this technology was initially published in Science back in 2015 and has been highlighted as Method of the Year by Lecture Methods back in 2020. And it truly opens up a lot of application areas. For example, you can profile the gene expression in situ across very large tissue areas and characterize cell function and state, analyze the intracellular organization of the transcriptome, and map the cell types in complex tissue. So in the next few slides, I'm going to highlight the key application areas, and then I will go through some of the advantages of the platform as well. The first application is to profile gene expression in situ from the whole tissue down to subcellular resolution. So shown here is image actually with a panel of 500 genes imaged in human ovarian cancer, and on the left, only 8 selected genes are shown here to really illustrate how you can use this to profile different marker genes in tumor samples. And overall, around 174 million RNA transcripts were detected. And I'm going to play a video on the right where you can really see first these 8 genes in display, and so here it is. And then as we zoom in at 2 single-cell resolution, then we turn on all 500 genes, and hopefully you can really see the richness of the data at single-cell level. And then as we zoom out, and then I'm going to turn off the 500 genes, and then only show the 8 selected genes. And hopefully this gives you a very quick glance of what the raw data coming from Merscope looks like. So leveraging the gene expression profile at single-cell level, the next key application that you can now imagine is to sort of characterize the cell types in complex tissue. And this is a publication coming from Science back in 2018, where the researchers used a panel of 155 genes and they imaged the mouse hypothalamus regions. And specifically, using MERFISH, they were able to identify 75 different cell types. And as shown by this t-SNE plot here in the middle, all the major cell classes were shown in the t-SNE plot here. And by benchmarking with single-cell RNA-seq data, hopefully you can see from the correlation plot here, MERFISH was able to recapitulate all the major cell types in the brain, indicating that this is a technology that can really reproduce some of the findings by single-cell RNA-seq. However, it does offer things that single-cell RNA-seq cannot offer because it's a spatial measurement, and we do have the coordinates of each individual cell. So at the top right now, what you are seeing are each color dots are now individual cell types, and hopefully from anterior to posterior, you can see the distribution of different cell types in the mouse brain. Furthermore, by including marker genes that can report neuronal activity, now the authors included a specific gene called CFOS. They were able to identify specific cell types that are responding to social cues. In this example, for example, parenting behaviors or mating behaviors. And hopefully you can see that some of those neuronal clusters that are shown in this red bar, they're responding to those social cues very specifically. So this truly highlights the power of using MERFISH to do true cell atlasing. So now with the cell type identified, the next application now we can think of is how different cells interact with each other. On this slide, this is a publication coming from Cancer Cell last year, where the researchers used a few hundred genes and imaged glioblastoma sample, and they were interested in how macrophages interact with the cancer cells. First, again, they benchmarked the technology versus single-cell RNA-seq, and hopefully you can see from the heat map plot here, MERFISH was really able to recapitulate what single-cell RNA-seq was able to find. But since, again, it has spatial information, MERFISH provides more information, and specifically, they measured the mean number of macrophages in 30 nearest cells to each MES-like glioblastoma cells, and they found enrichment of macrophages adjacent to this subtype of glioblastoma cells in situ. So you can imagine, without a technology like this, such a finding will almost be impossible. So in the next few slides, I'm going to highlight a couple of key advantages. First, this technology is a very quantitative measurement, and you can measure it across a very large dynamic range of gene expression. Showing here, this is on the left is a figure coming from a PNAS publication back in 2016, where we benchmarked the sensitivity versus single molecule FISH, which often is considered as the gold standard for quantifying RNA copy numbers. And from here, you can see that MERFISH was able to recapture around 95% of what single molecule FISH found. So it's a truly sensitive technology. And importantly, this sensitivity sort of spans across a large range of expression level. On the right now, this is in tissue mouse brain imaging. Around 500 genes were imaged, and each dot is a single gene here. Y-axis is the copy number per cell measured by MERFISH. X-axis is the FFPE from bulk RNA sequencing. And hopefully you can really see that not only MERFISH was able to detect genes at high expression level, but also down to 0.01 copy number per cell. And it maintains a very linear correlation with correlation coefficient of 0.88 here. Secondly, this is a truly sensitive technology, and on this slide, what I'm showing here is two publicly available data sets coming from, one is coming from an array-based platform, another way of doing spatial genomics measurement in matched brain slices. And a single gene was highlighted here called OPRD1, which is a GPCR. And hopefully with Murphy's data, you can really see where OPRD1 is in the brain. However, as shown at the bottom, you can see with array-based platforms, there are only a couple grid spots showing here. And if we zoom in, you can see which individual cells expressing OPRD1, for example, in the hippocampus region there. However, with grid size of 55 microns and in between there's dead space, it's not really true single-cell resolution. And when we compare these two data sets, we found that Merscope consistently detects 70 times more transcripts per gene versus the array-based platform. Next, this is a very high cell throughput platform. So as an imaging-based platform, you can imagine in order to increase the cell throughput, all that we need to do is to scan more areas in tissue. And specifically for Merscope, we can now profile up to a square centimeter-sized tissue with hundreds of thousands, sometimes more than 1 million cells captured in a single experiment without any downstream sequencing need. So that really allows some very rare cell types to be easily captured on our platform. Next, this platform is also compatible with protein co-staining. We have recently, earlier, I think this month, released a protein co-staining kit where you can now overlay proteins on top of RNA. Showing here is 5 protein staining in the mouse brain and with hundreds of RNA stained, which is shown on the right. And hopefully you can really see the beautiful morphology highlighted by the proteins. And now this really pushes the technology to a spatial multi-omics approach now. And finally, the technology has been very rigorously validated in the field. Since the initial publication in Science back in 2015, the more than 40 peer-reviewed publications and preprints, many are in Nature, Cell, and Science, and many high-profile journals. And many institutions around the world are already using Murphy's and Merscope through Wisdom's commercial efforts. So we launched the platform, actually did a soft launch back in August last year, and earlier this year in January, a full launch in the US. But the first chemistry that we launched was on fresh frozen or fixed frozen samples. Then it really bears the question, like, can we work on FFPE samples or not? So in the second half of my presentation, I'm going to highlight how we can use Merscope to profile FFPE samples. And I'm going to use this slide to sort of quickly walk through some of the pros and cons of different tissue preservation methods to really highlight why there's such a critical need. As you can imagine, fresh frozen samples can sort of better preserve the DNA and RNA, and oftentimes they're compatible with single-cell RNA sequencing analysis. However, this is not a very common sample type in clinical space, and sometimes you just cannot find such sample types. While on the other hand, FFPE samples are widely available, and they store at room temperature, very easy to handle as well, and it could better preserve the tissue morphology. However, this is a more challenging sample type to work with, especially when you wanted to profile RNA or DNA, because there's a substantial cross-linking happening in FFPE samples. And degradation and fragmentation also makes the probing of RNA much more challenging in FFPE sample types. So overall, it requires a substantial change of chemistry in order for a technology to work for in situ single-cell transcriptomic imaging. So within Wisdom, we have now developed a new proprietary chemistry to enable this in situ single-cell genomics imaging in FFPE samples now. And I'm going to use the rest of the slide to really highlight how we do it and what the performance looks like in FFPE samples. First, to establish the performance of our chemistry, we chose mouse brain samples to benchmark the performance because in mouse brain, it has very well annotated, publicly available in situ gene expression profile information. So shown here now, this is a FFPE mouse brain imaging with a panel of 483 genes. As you can see from here, we showed the distribution of all transcripts and a zoomed in single field of view here. Across this single piece of tissue, we were able to identify around 45 million transcripts in around 55,000 cells, and each cell has an average around 816 transcripts. This is similar to what we found in fresh frozen mouse brain imaging, datasets that we have posted to the public earlier last year. And to benchmark the performance, we have in the bottom now chose a couple of genes, 4 genes here, and compare that with Allen Institute's ISH data. And hopefully, as you can see from here, all the genes showed the expected distribution as compared to Allen ISH. And of course, you can check all the other genes as well, and it matches very well. To more systematically benchmark the performance of our workflow now, on this slide, we're going to do a couple more rigorous benchmarking. First, by comparing the data coming from our FFPE workflow versus bulk RNA sequencing. And showing here now is the comparison of the counts coming from Merscope versus the FFPE value for the same genes measured by bulk RNA-seq. And each dot is a gene here. And hopefully from this correlation coefficient plot here, you can see that it's a highly quantitative and accurate measurement with correlation coefficient approaching 0.88. And then it's highly reproducible. This correlation coefficient is 0.999 something, so it rounds up to 1 here. But in between biological replicates, it's highly reproducible. Next, we also wanted to benchmark the chemistry that we have released already, which is the fresh frozen workflow. So we performed the Merscope imaging in fresh frozen samples using the fresh frozen protocol versus FFPE samples with the FFPE protocol. And now, again, showing a correlation coefficient 0.95, and it really indicates it has comparable sensitivity to our fresh frozen workflow. Furthermore, since we do have the ability to do single cell analysis, the last benchmarking that I'm showing here is to sort of cluster cell types, identify each individual cell type, and then map them in space. And again, hopefully you can really see with FFPE workflow, we were able to identify the same cell types, and then the location of those identified cell types matches very well with fresh frozen samples as well. So now, hopefully these 2 slides sort of set a stage and establish the performance of our chemistry. Then the next question is, can we apply this chemistry to other sample types, and especially in human sample types? And this is what this slide is showing now. We used a 300 gene panel and measured more than 12 different FFPE clinical samples. And for example, ovarian cancer, lymph node, tonsil, lung cancer, human skin, and such. And hopefully these are a couple of genes highlighted out of this 300 gene panel measurement, because it's very difficult to show 300 color at the same time. But hopefully with these marker genes shown in these images, you can really appreciate how we can now profile biomarkers in samples like this. And I wanted to highlight, for example, skin samples. If we wanted to do single cell analysis, it's extremely challenging because it is a sample type very fibrotic, has a lot of collagen. Thereby cells are very difficult to dissociate. But with an in situ measurement like this, all that you need to do is to slice the sample and hybridize the probes, and then you can image it. And now you can see where keratin, vimentin, and fibroblast markers such as ACTL2, where they are in space. And of course, there's always the question how accurate this measurement is. And for all the measurements, we did a bulk RNA sequencing in the sample block and compare that with the Murphy's counts. And again, with the correlation coefficient 0.81 here, this is for one of the cancer sample types. Hopefully you can really see that it's quite accurate, and we did this across all samples and just take the R value from the correlation plot here and plot it in bar chart, and they're all above 0.7, so indicating that this is a very accurate measurement across all the sample types we tested. Furthermore, the workflow is still compatible with protein co-staining. So in this picture here, what I'm showing is FFPE human colon cancer with a panel of 347 genes and 6 proteins co-staining. And pan-CK, SMA, E-cadherin is shown here, and around 67 million transcripts were detected. And hopefully from the zoom-in images, again, you can see the beautiful morphology stained by the protein co-staining, and now the overlay on the right with the RNA on top of the protein. Finally, this FFPE workflow enables true single-cell identification because we do have a cell boundary kit to label the plasma membrane of the cells. And with that, then that allows us to segment individual cells and identify where they are in dense tissues. In this particular example, it's a human lung. On the left, this is imaging 244 genes. On the left, this is showing about 6 genes as a representative image here. You can see this very beautiful human lung imaging. And with that cell segmentation algorithm, we were able to classify different cell types in the human lung. And in the zoom-in image here, hopefully you can really see, for example, with AGER positive type 1 cell and SFTPA2 type 2 cells, and The pink cells are the macrophages, and then we have fibroblasts surrounding the bronchial-like structure labeled by MUC4 over them. So hopefully you can really now, in a complex tissue like human lung, really see some very, very beautiful structure like this. So in the last sort of set of slides, I'm going to highlight now with the capability of FFPE imaging established on Merscope platform, what you can do with this technology. And I'm going to highlight just one single case study doing cancer cell atlasing. And this is data that Wisdom has released earlier this year. If you're interested in downloading, this is a link here. But essentially, this is 500 gene focused on immuno-oncology, focused gene panel, profiling 8 major tumor types, including colon cancer, liver cancer, melanoma, ovarian cancer, prostate, lung, breast, uterine cancer. And 16 datasets were acquired, and more than 4 billion transcripts were available in these datasets in around 9 million cells. We believe this is so far as we know, the largest single-cell spatial transcriptomic datasets publicly available in this kind of format. So on the right, again, in order for us to really build the confidence of how it works, we benchmarked our technology versus traditional technologies such as bulk RNA-seq again, and hopefully in the middle you are seeing the bulk RNA-seq correlation plot again now, and this is showing, the plot is showing the ovarian cancer, and you can see correlation coefficient is 0.82, and in between biological duplicates, it has 0.99 correlation coefficient, and across all samples, it all has correlation coefficient above 0.7. And separately, with the single cell resolution and the ability to segment cells, we were able to classify different cell types using UMAP clustering and then map out where individual cell types are in space. And at the bottom now, each colorful dots indicate a different cell type we identified across these 8 different sample types. And hopefully from these very pretty pictures here, you can really appreciate the heterogeneity of cell types in cancer. To illustrate what kind of biological findings that you can get from such data sets, I'm going to zoom in into one data set and really highlight some of the biological findings and insights. And this is on a triple-negative breast cancer here, where first on the left, what you're seeing is a UMAP plot of the different cell types identified in the breast cancer. We were able to identify 14 different cell types, major cell types in the breast cancer cell type, including, for example, you can see this very big bottom here. This is a fibroblast cluster, 2 population of fibroblast over them. And at the bottom, this is a dot plot showing the marker genes for each identified cell types. And hopefully you can recognize some of those markers, for example, for T cells or for endothelial cells. So in single-cell RNA-seq, sometimes when you try to identify cell types, due to the variations of cell dissociation, sometimes there's this uncertainty that caused by, say, dissociation protocol or doublets. How do I know, for example, that a cell type is a true cell type, right? For example, in this UMAP plot, we have 2 fibroblast population. One is this big green cluster here, and at the bottom, there is a very tiny red cluster of fibroblast. When such a UMAP plot is shown, like, do I know for sure whether that's a fibroblast population for sure or not? And now with the imaging-based platform, you can actually zoom in into the data sets and really figure out whether that's a true cell type or not. And we did that with these data sets. And we zoomed in, for example, in a region of breast cancer. And in figure D now, you can see the 2 population of fibroblasts that we were able to identify. Hopefully you can see from this single field of view image here, both were expressed in the hallmark gene of fibroblast called collagen 1A1. But interestingly, the second population, the rare population, is expressing a gene called MKR67. This is a cell proliferation marker. So this kind of really illustrates that fibroblasts are displaying 2 unique states. One is more actively proliferating, while the other is not. And now it really highlights the power of an imaging-based platform to not only identify cell types, but also characterize cell states as well. And in this sample type, we have identified a lot of immune cells. And in cancer biology, especially in immuno-oncology field, oftentimes one of the questions is how different immune cells are infiltrating into the tumor. So taking the green cluster, which is at the top right here, this cluster, this is the immune cluster here, we're going to zoom in and do a subclustering analysis. And that further that allowed us to identify 7 more subtypes of cell type, immune cell types, including, for example, shown in the marker gene in figure B now, myeloid cells, regular CD4 cells, CD8 T cells, T regulatory cells, and NK lineage cells. So you have all the cell type annotated now, but also at the same time, you know where those immune cells are. For example, showing at the bottom now, this is T regulatory cells and CD4 cells. Hopefully you can really see that T regulatory cells are more infiltrating into the tumor, while the regular CD4 cell types are more restricted at a region where a lot of endothelial and blood vessel structures are. And furthermore, you can zoom in into the data sets and really visualize where those cells are and how they express those marker genes. And in this single field of view image here, hopefully you can really see now, for example, T regulatory cells expressing a hallmark gene called FOXP3. At the same time, exhaustion marker CTLA4 in white now is also co-expressed in these Tregs. And at the same time, you're seeing a couple of NK cells expressing the hallmark gene called NCR1. And then, of course, we have CD4 and CD8A marker genes expressed in other cell types as well. So since the launch of the products, we, of course, have a lot of other applications and different examples. And given the time, I want to be able to really go through all the examples. But I wanted to highlight in this single slide some of the publications that we already have. This is still not the complete list. There are many more out there. But I wanted to highlight actually just in 2022, there are already around 20 publications. And to highlight some of the key advantages and advances in recent weeks and months, one of the studies published in Science was a measuring of 4,000 genes in human brain using Murphy's technology, now truly pushing to near transcriptome level. And about a few weeks ago, there is also a publication in Cell, actually doing spatially resolved epigenomic profiling, now truly enabling us to a spatial multi-omics measurement now, because now you can see epigenomic modifications in situ. So this is a publication in Cell, I believe, come out about 2 weeks ago. And lastly, just sort of highlight some of what our customers are saying. For example, this is from the VP of Allen Institution, Hong Kui, where they were doing a lot of brain initiative studies to map out where individual cell types are. And they found that this is a truly powerful way to characterize different cell types, not just in the brain, but in many other parts of the body. And recently, we have also have another site coming from TGEM. Actually, again, they compared our platform versus many other platforms, and they found Merscope captured significantly more single-cell data than the other instrument. And TGEM Nick actually is here, and he has a poster, 3179 here. They were using this to image a diseased state of human lung, and if you have time, highly recommend you to check out the poster over there. And just to wrap up everything here, so just to summarize some of the key highlights of what Merscope's advantages are. First, this is a truly multiplexed platform. It can cover hundreds to even thousands of RNAs in a single sample, and it's completely custom. And for now, we offer custom gene panels, but we also have some pre-designed gene panels, standard gene panels to be released very soon. We also have protein code detection capability. The platform has very fine resolution from whole tissue to single cell to subcellular resolution down to 100 nanometers. It has truly exceptional sensitivity and specificity, actually the highest sensitivity, detection efficiency for identifying RNAs, has very high cells throughput in basically can capture hundreds of thousands cells in a single sample without any need of downstream sequencing. And it's a truly flexible platform that are available to run almost any sample type, such as cultured cells, suspended cells, fresh frozen samples, fixed frozen samples, and FFPE tissue blocks. With that, I wanted to say thank you, and if you have any questions, please let me know.